Well, thank you, the organizers, for the invitation. For today's presentation, I would like to describe basically what has been our experience when transferring manufacturing process from academia to a private sector, and describe what are the challenges and opportunities related to this. But first of all, let me say something about Marker Therapeutics. Marker Therapeutics is a clinical stage biotech company located in Houston, Texas. And the purpose of this company is really to advance multi-tumor-specific T-cell products that have been licensed out of Baylor into the clinic and future commercialization. At the moment, the lead product of the company is called MT401. This product is a multi-targeted T-cell product that recognizes 4 different tumor-associated antigens. And I will be describing that later on in more detail. And this product is geared towards being tested clinically in patients with AML post-transplant. And at the moment, that is our lead clinical indication. Now, when we look at Marker Therapeutics, at the moment we have, as presented in the slide, management team of sufficient experience in the transition of products into the commercial setting. And at the moment in Houston, we have developed an infrastructure that will allow the execution of these studies in the clinic and future commercialization. As shown here in this slide, at the north of Houston, Texas, we have constructed a GMP facility that will serve to manufacture products for this phase 2 clinical trial in patients with post-transplant AML. We also have a contract and supply agreement with Baylor College of Medicine, with the GMP facility for the Center for Cell and Gene Therapy, where we can also use some of this infrastructure to manufacture product for the clinical trials. And as shown in the lower left-hand side, the company also have a research laboratory where some of the process optimization, as well as the immunomonitoring to support the execution of the clinical study, is also conducted. And finally, the headquarters of the company is highlighted in the top left-hand side, where the clinical operation, the clinical regulatory team works to coordinate a lot of these activities related to the execution of the clinical studies. Now, but if we go into a little bit more detail in terms of what multi-tumor-specific T cells are, this product is a natural product that is not genetically engineered, which is generated via the stimulation with overlapping peptides using professional antigen-presenting cells to expand a polyclonal population capable of recognizing multiple epitopes and multiple antigens simultaneously. The final product is constituted of thousands of individual alpha-beta TCRs. And in this way, this natural product has really a very broad spectrum target recognition. Now, unlike traditional monotherapy or CAR T cells that are able to recognize a single epitope of a single antigen, this normally, although effective at first approach with the cancer cells, this normally is related with a subsequent tumor relapse, as the tumor cells that do not express those antigens are able to escape that immunological pressure. Well, in contrast, as shown here on the right-hand side of the slide, our therapy is a multi-targeted T-cell approach that is capable of recognizing cancer cells simultaneously. In addition to this, our therapy is given to patients without the need for lymphodepletion. And this also allows the recruitment of the endogenous immune system through a phenomenon called epitope spreading. So what we see here is, in fact, 2 waves or 2 mechanisms of anti-tumor activity. The first one derived from an initial killing derived from the cells that we infuse to the patients. And then the second one derived from this induction of the immune system that increases the breadth of immune response even further. Importantly, this product can be given to patients And at the moment, we have treated over 150 patients with this technology across of different clinical trials, including hematologic malignancies and solid tumors. And we have seen a very favorable safety profile with the identification of objective clinical responses in some cases. And again, I'm going to highlight some of those clinical responses in a moment. But one of the things that is very important is the safety profile of this therapy that allows, first of all, the administration of these cells in the outpatient setting. So patients can come in, the product is administered, and they are monitored for a couple of hours, and they can be sent home. In addition to this, the safety profile, the favorable safety profile of this drug also enables the use of this product in the adjuvant setting, right? So this can be given to patients that are currently in remission in order to maintain those patients in remission for a longer period of time. Now, but let me dive now perhaps into more technical aspects in terms of how the process actually was at the moment that we did the initial license from Baylor at the time of the transfer of the technology. This slide illustrates the Baylor manufacturing process the way that it was at the time of the license. This manufacturing process relies on multiple rounds of antigen stimulation, expanding cells in 24-well plates, requiring multiple handlings of the culture to produce a propagation and administration of growth factors and media. And even though we have a significant clinical experience with a good safety profile and evidence of clinical efficacy, still the manufacturing process was really basic, as illustrated here in this slide. Now, what I can now show, and I will explain in more detail how we got to this point, we are now to a stage in which the manufacturing process has really evolved from that complex stage of development into something that is now a lot simpler, right? So now we're talking a shorter in vitro culture process. The T cells are expanded in a platform called the G-Rex, which basically allows cells to grow undisturbed for a long period of time. The configuration of the G-Rex, which has a gas permeable membrane on the base, allows the administration of large volume of media up front. What this results is in the expansion of T cells without the need for media exchange or agitation of the culture, minimizing all the unnecessary steps and manipulations of the culture throughout. And this has been significant, very important, as we basically now have simplified the manufacturing process of our lead product, MT-401. So we have gone from a process that was really complicated into something that now we will consider simple and robust and appropriate now to take into the clinic. And I think that the word of caution, basically it comes down to provide a parallel between the challenges and the opportunities. I would say that although investing sufficient time, and the previous speaker alluded to that, in the manufacturer is really an investment, I guess doing this effort as early as possible is really, really important so that you have that uniformity on the manufacturing process as you go into the clinical developmental program, so that you want really all your patients, or the vast majority of your patients, to be dosed or treated with a process that really provide a very robust platform. And with that said, I think that most people will agree that there will be room for process improvement. However, having a very solid foundation of the process allows for these future changes to be a lot more gradual and allow for adaptation without necessarily being considered a new product and putting into question the uniformity of the clinical data, right? Now, when we look at the characteristics of the final product, we basically see here, and before I get there, let me highlight what is shown here on the left-hand side of panel A. The reason we went on this direction of investing in really thousands of hours into manufacturing process optimization to simplify the process was really to show what is illustrated in panel A, which is to minimize the number of interventions and to simplify the production of the final product, right? Now, although that was the main objective, what we actually learned, and this was a byproduct or a surprise out of that event, is what is shown here in panel B and C, and is that as we actually were able to improve the manufacturing process and simplify the number of the interventions, we actually were able to learn that this improved manufacturing process also led to an improved cellular phenotype, as illustrated here by cell characterization that we will consider to be superior with the product that it was licensed out of Baylor. In addition to the improved characteristics of the cell phenotype, we also observe here an improved distribution of antigen specificity. So here you have the ELISpot, which is the method that we use to measure the specificity of the final drug product. And here you have in the axis the number of spots forming cell by an interferon gamma ELISpot, and you have 3 different donors. And what you can basically see here, if you look, for example, at donor 1, is the distribution of antigen specificity in this new manufacturing process that is shorter, results in a product that has a broader spectrum of antigen specificity, which would also will speculate would actually result in a broader target properties of the final product. And hopefully we'll be able to see those results in the near future in the clinical studies. And the product profile that we are able to generate looks something like that. So we're able to see that the cells expand throughout this new manufacturing process, and we have a final product that has both CD4 and CD8 T cells, and a balanced distribution of central memory and naive T cells, and a robust representation of antigen-specific T cells at the end of the culture. Okay, so let's take a small break on the manufacturing process. And let me now jump back into the clinical data. And I guess the point of this is I'm going to highlight some clinical data that is really studies that were developed out of Baylor College of Medicine, right? And I'm going to then go back to the developmental history of the program. So as I mentioned before, one of the things that was really unique out of this program is that when we initially performed the license out of this technology from Baylor, really was significant clinical data that shows the product to be safe and some evidence of clinical efficacy. But really the biggest roadblock is the state of the manufacturing process, as I just highlighted, right? And I guess since the moment of the license as presented now, we have been able to significantly improve the state of the manufacturing process. But let me show you in more detail what some of that clinical data looks like. So what we basically are shown here, this is data that it was generated out of Dr. Ann Lin's lab at Baylor College of Medicine. And you basically see here, these are the clinical responses of patients enrolled in a lymphoma study treated with multi-tumor-specific T cells. And you see here the arrows in a swimmer plot changing from blue to green. You see that those are basically highlighted as the complete responses. And one of the things that would be important to note is that these patients received this as a single therapy. And importantly, you start seeing this change of complete response that is quite actually late in the treatment, normally anywhere between 6 to 12 months after the administration of the multi-tumor-specific T cells. So I think that when you look at this data, this is a clear signal that this therapy as a monotherapy is able to actually work and provide objective clinical responses, but you basically see a kinetics of clinical efficacy that is actually quite late or delayed. In addition to this, we also see now, this is the similar swimmer plot, but now showing the results in adjuvant study. And this is only possible due to the good safety profile that we see out of this product when given into patients. And of course, the caveat of these results are that this is a single-arm study, and of course, these patients are in remission at the time of receiving the T cells. But what you can basically appreciate is that at the moment that the patients are enrolled and after receiving the product, these patients remain in remission for a sustained period of time, surpassing what it is expected from patients that have a similar diagnosis, right? So is something that is highly encouraging of how this therapy actually could also work not only in patients with active disease, but also in the adjuvant setting. But now moving from lymphoma to AML, which is the lead indication that Marker is advancing into the clinic. Here I'm showing data also from patients enrolled in an adjuvant study, and you see a very similar profile, which is patients that are treated basically surpass what it is expected from patients similar staging. And if we look now at patients with active disease, we basically also observe some evidence of clinical responses here as complete response or partial response in a clinical condition that is highly aggressive. So due to all this clinical data, Marker Therapeutics have decided to move forward into the clinic as a first indication, a product that we call MT-401. It's a product that has multi-targeted specificity and is focused for the treatment of patients with AML post-transplant by treating both patients that have active disease after a bone marrow transplant, as well as patients that are in remission, so on the adjuvant setting as illustrated in these slides. And let me walk you very quickly through the layout of the proposed clinical study. As shown here, this is there by definition an allogenic product, but it's a patient-specific allogenic product. So patients that have AML receive a bone marrow transplant, and after the patients have relapsed, or after 90 days, these patients are then enrolled in the study, and at that point, the product is collected. So you have the enrollment of the patient, and then the procurement of the donor of the bone marrow transplant, which material is then used and sent to the GMP facility to expand and produce tumor-specific T cell product, which is then cryopreserved. At this point, these cells are then sent to the clinical site when they can be administered. And what is shown here on the right-hand side, you see that after this, these patients are monitored periodically by peripheral blood collections to evaluate the frequency of multi-tumor-specific T cells in peripheral blood, as well as correlating this with the clinical performance. And this shows the layout of the clinical study in a little bit more detail. So we basically are, as I mentioned before, we have two main groups. Group one, which is patients in the adjuvant setting. In this group of patients, the patients will be enrolled and then randomized at a 1-to-1 ratio, receiving multi-tumor-specific T cells or a standard of care, versus patients in the active disease group, in which they will receive tumor-specific T cells alone, and this will be a single-arm study that will be then compared with real-life historical data. But one of the things that we wanted to see is, okay, so we now are starting into this clinical study. We already finished the safety lead-in where we're comparing two different materials, and at the moment we're in the mix of the phase 2 portion of this clinical study. And I think it was definitely really important to first improve the foundations of this process that we use to generate the cells. But where do we go from here, and what are the next things that we can actually do to further improve the manufacturing process? So I'm going to show something that we have done recently and we are incorporating into the manufacturing process, and it's the incorporation of the Lovo system. And this is perhaps the most important slide, so I'm going to spend a little bit more time here. So what you can basically see, and is shown here on the right-hand side, is when you compare the cellular yield by a manual cycle or Lovo, you basically see that you are able to pretty much double your cellular yield by using the Lovo. in addition to that, you also further simplify the manufacturing process by decreasing the processing time from 3 hours to only 1. And you're now using a process that would enable the manufacture of the cells in a closed system. And using the foundation of a simple and robust manufacturing process, now this can be incorporated to further reduce the number of manipulations in the generation of the cells. This just shows that when you look at the composition of the cellular yield, there is a greater content of lymphocyte, which in our case is actually beneficial, given those are the cells that we actually want. And when we compare the MT401 that is generated with manual FICO versus the Lovo, we basically see that at the onset, we see that the yield is different, but once you actually set up the culture, you're able to see that the MT401 fold expansion is comparable, whether the product is manually isolated by FICO or Lovo, and you also see comparability in the cell viability. And in this case, we're able to see a comparable phenotype when we're looking at the distribution of both CD4, CD8s, as well as the effector and centromere profile of the final product. And finally, we see a distribution of antigen specificity that is comparable. So in conclusion, what you can basically see here is that by incorporating the G-Rex and the Lovo, we have been able to dramatically simplify the manufacturing process. Some of these improvements, as shown in the earlier part of the presentation, not only were able to simplify the manufacturing process by reducing the number of interventions and the days of culture, but also resulted in a product that had superior characteristics when looking at the cell phenotype and antigen specificity. And I think that this will create a solid foundation as we now move forward into the phase 2 portion of the clinical study. But looking into the future, I'm just going to finish here with something that is really more than anything, is just really food for thought. And is one of the work that was recently presented at a different meeting by one of our colleagues, Dr. Anastasia Smith. And this work shows our current collaboration with ABB Robotics, in which we're now incorporating a system that would allow us to further automate the manufacturing process. And I think that this is something of high interest, and we're really excited about this collaboration. And the reason for this, and the reason I think this now is even feasible, is the fact that you're having now a very simple manufacturing process that will actually enable a robotic assistant to come into the laminal flow and help the operators in the GMP with some of these manual tasks. So please stay tuned. We will continue updating as we perform additional advances in this area. And with that, I would like to finish. And if there's any questions, we'll be happy to take them. Thank you.
Well, thank you very much, Juan, for the very interesting presentation, and certainly exciting wrap up there of looking at getting that robotic conversion over to the G-Rex. I think a lot of folks will be interested to see that that one get to the next stage. I think the first question that's come up from the group is, in the clinical data, when you know the relapse, is that meaning that they were moved on to different treatment plans, or did you do another dose of the multi-TAA product?
Okay, so and just to clarify, these are patients that come out of relapse out of the bone marrow transplant, right? So prior to the bone marrow transplant, these patients have received, a lot of other line of therapies, right? And in the clinical paradigm, I mean, bone marrow transplant is one of the last resources, especially an allogeneic transplant is really one of the last resources for many of these patients. So these patients that will fail the bone marrow transplant, in that case, is where they become eligible to receive MT401, right? And at that point, the therapeutic options are really dismal for this group of patients. So I think that any change that we see in the clinical front there will be quite significant.
Okay, great. And when you look at the MT401 phenotype data, when you look at variability, for example, CD4, CD8 ratios, Is there any variability in patient outcomes based upon that 4:8 ratio that you've observed?
Yeah, that's a great question. I think that especially in the CAR field, there is a lot of awareness of the importance of the constitution of both CD4 and CD8 products, right? And I think that there's definitely an inherent effect of the process that changed the constitution of the phenotype. What I have to say is that variation that we see is mainly derived from the starting material itself. If we repeat the same process multiple times with the same starting material, the phenotype is spot on. So we get the same distribution of both CD4 and CD8. So it's basically a variable that is at this point outside of our control, as really is related to the incoming material. Now, to address your question, we have looked into the data. And I don't know if it's simply because the data set is so far very small, even though we have about 150 patients at the Baylor studies, but so far we have not seen a correlation in terms of the proportion of CD4, CD8 cells and the clinical outcome.
Great. So I guess continuing kind of that theme of thinking about, manufacturing specs and variability and clinical correlates, what would you say is your overall regulatory strategy for knowing, obviously, potency being such an important thing on these products, right? What would you say is kind of the regulatory strategy for the potency release for the multi-TAA therapy? By each antigen, or are you thinking total response, knowing that there will be some variability, of course, for whether or not it's WT1 or PRAME or other things in there?
Yes. I mean, obviously, that is a field of high interest, particularly when you talk about a product such as this, that is a natural T-cell product that, due to the polyclonality and diversity, is sometimes harder to characterize, right? Now, we are using as a parallel sort of the experience derived from virus-specific T-cells, right? Which it has been in the clinic far longer than tumor-specific cells, right? So if we use that as a parallel, what we basically see is that at least a mechanism that has been accepted is the use of interferon gamma production. And what we have here in our advantage is the use of the unknown source of stimulant, right? So I think that unlike TILs, for example, where the antigens that are used to stimulate the product is unknown and is very variable. You now have two variables, the variability of the starting material from a T-cell part, but also the variability of the tumor. Is harder to control. In this case, at least we have the control on the antigens and the distribution that are used to stimulate the product. So what we're now looking for is for the production of interferon gamma and the accumulative production of interferon gamma in response to that. And I think that will be our regulatory strategy moving forward. But hopefully that's something that with experience from the field, we'll be able to circumvent without much delay.
Great. All right. And the last one we have up here is, wondering what the dose for the multi-TAA and the production yield of the manufacturing process that you've kind of taken forward at this point.
Oh, yeah, that's a great question. I actually didn't mention that in the presentation. What we're now exploring is a flat dose of 50 million cells, which is given to patients 3 times intervals of 2 weeks. And at the moment, we're actually in the middle of a dose escalation. That's a study that it has been conducted in collaboration with our colleagues at Baylor. And now we're looking into increase this cell dose beyond what we currently are doing. I don't think we have publicly actually presented what those numbers will be, but we're currently exploring into increasing those cell doses. And I think that it's sort of just expected to basically be able to see a correlation in terms of dosing and clinical efficacy, especially when you're treating patients with active disease in a disease that is highly aggressive and very rapidly growing like AML.
Well, thank you for all of the great responses there and for a very nice presentation, Juan. We appreciate all the insights you've given today.